This video looks at species and reproductive isolation, and is part of maintaining life, and especially the evolution part of that. So one of the issues facing biology for a couple of hundred years even has been, what is the definition of a species? And we'll take you through some of the different ways in which species have been defined, and look at reproductive isolation, which is a main key way that it's defined in sexually reproducing species. So initially, when Carl Linnaeus was wanting to develop his system of taxonomy that we still use today, the Latin binomial system, he used morphology as a key way of looking at which species looked similar and had similar sizes and shapes and patterns in order to determine which ones are similar species. Although this has been very successful, and obviously members of a species tend to look very similar, it can also lead to some problems. So for instance, here we might look at these 4 different butterfly and try and work out, are they 2 species? Are they 4 species? Well, it actually turns out that these represent 3 different species. And the way in which that works is actually we have 3 species here, and they have seasonal forms. So the colors on them, these are the same species even though they look quite different to each other, whereas these ones are very similar and they're actually in different species. And so sometimes colors, patterns, even size and shape aren't a very good way of working out which species are the same and which are different. However, these morphological differences are very useful in other aspects of evolution. So for instance, if we look at the bone structure in the arm, leg, flipper and wing of these 4 different mammals, we can see the same bones in terms of the upper arm, radius and ulna, wrist bones and finger bones conserved across evolution. Even though we have a bipedal, quadrupedal, swimming and flying mammal, they show their relatedness through these morphological characteristics. So morphology alone isn't very reliable as a way of defining species. It is very easy to do and is commonly used, but its main power is in looking at relatedness on a broader scale and as part of other evidence. Something that's perhaps a little bit more useful is biochemistry. So even though animals might look the same, another thing that they have is their internal biochemistry, which can operate in the same way, or the way the cells and organisms function, which we call physiology. So their similar physiology is a really good indicator of their relatedness. Although this diagram might be complicated, it's a relatedness between some different groups of organisms, and in particular these types of proteins called beta defensins, which have evolved from being harmless to being venomous. And so venomous versions occur in lizards, snakes, and even in the platypus. And so we can see that the evolution of venom in these populations has been derived from a similar protein, and so we can track relatedness by looking at those similar proteins, in the same way that related organisms might have similar enzymes which allow them to have similar diets, or be salt tolerant if they're plants and things like that. So the biochemistry, the way the cells and organisms function, gives some connectedness to their relatedness, and in some cases can help to define species. More commonly and more recently, gene pool is used as a way of defining species, but this is only really that useful for sexually reproducing species. So each member of a population, of course, in a sexually reproducing species will have homologous chromosomes which form their genotype. In this case, we're looking at one location on the chromosome, or locus, which is X, that has 3 alleles, 1, 2 and 3. So the gene pool is the full collection of all those different alleles within that gene. And so because any members of these species could sexually reproduce, if they're male and female, then we can mix up and share those 3 alleles across all members of the population, and therefore they're all considered to be part of the same gene pool. Because they're all part of the same gene pool, then we also call that part of the same species, because they can interbreed and produce fertile offspring. And so one very common definition for species in sexually reproducing organisms is that they can successfully interbreed to produce fertile offspring. And that means that they all end up being part of the same gene pool, because they can share their genes within that population. Another good way of understanding this is to look at what are the barriers to reproductive isolation. So what puts organisms outside of this gene pool and therefore in a different species. So here's a whole lot of what we call reproductive isolating mechanisms, some of them being called pre-zygotic, that stop interbreeding before fertilization can happen, and some called post-zygotic, which stop the production of fertile offspring after fertilization has happened. So for example, if two populations mate at different times of year, we might call that temporal isolation. So these two frogs are not part of the same gene pool because they won't breed with each other because they breed at different times. You could have ecological or geographic separation. Again, that would mean that the two don't interbreed even if they could. Behavioral isolation means that even if two groups or populations could potentially interbreed, then because they might have different mating rituals or different songs in the case of these birds, then they wouldn't interbreed, and so the courtship never proceeds. And the final one here is mechanical isolation, is that either through their reproductive organs being incompatible, or sometimes just the sperm and egg won't fuse, then that prevents fertilization from happening. But even sometimes, if all of these work out, that they're able to complete courtship and complete mating, and that the sperm and egg meet and fuse, that doesn't necessarily mean that they're the same species or will produce fertile offspring. So for instance, hybrid inviability means that some hybrids, although fertilization across two different species, will never develop into a full adult. I think this arrow is going the wrong way, but it's basically saying the tadpole will never develop into a frog because the two sets of genomes just aren't compatible, and although fertilization can happen, it will never fully develop into an adult. Hybrid infertility, the classic case of that is the mule. So even though a horse and a donkey can breed to produce a healthy animal, it is a mule, it is sterile and cannot further breed. So that's why the definition of species is that interbreeding is successful and produces fertile offspring. So here's the example of why that happens. So horses have 64 chromosomes, donkeys have 62, and so that when meiosis occurs, we've got 32 and 31 chromosomes that make a mule with 63 chromosomes. So it doesn't have pairs of homologous chromosomes. This doesn't prevent mitosis from happening. So mitosis can go forward and the cells here will grow and divide as normal. But it does stop meiosis from happening, because as we know, the homologous chromosomes need to align in tetrads for meiosis to happen. When you have an odd number of chromosomes like this, they can't successfully form tetrads, meiosis won't happen, and no gametes are produced.